Welcome to Life in Biology. I'm Dr. Joel Graff, and this episode is going to be talking about the current coronavirus pandemic as well as former pandemics caused by influenza. Just a little background: about a little over 4 years ago, I made a video about how scientists were able to resurrect the 1918 influenza virus. So they went out into nature, into archives, and found some sequence from the 1918 influenza. We're able to piece that back together in a lab, and at the CDC in Atlanta, they are working with 1918 influenza. It's in tissue culture in mice down there, and a few people have asked me about whether it's possible that the resurrected 1918 influenza could have anything to do with this current coronavirus outbreak, and the answer to that is no. But then people were asking if I would make just a little bit of a video to compare those two viruses, and so here, by popular demand, here we go. So on this image, I've got a rough drawing of the two different viruses that we're dealing with. On the left, we've got COVID-19, the virus that causes COVID-19. So this is the disease, COVID-19. The virus that causes it is called SARS-CoV-2. And the 1918, or Spanish flu, was caused by the 1918 H1n1 influenza. I have Spanish in quotes because this viral infection actually started in the United States, but somehow along the way it got the name Spanish. Anyway, we have COVID-19 virus here, the SARS-CoV-2, and I've drawn this before. I'll link to the video where I discussed this before, because I have a couple videos about how we can stop coronavirus replication. I'll also link to the video about how we got the 1918 influenza. That'll all be down in the show notes. But anyway, just as a reminder, or if you haven't seen that previous video, this virus has an envelope which is designated by that black line that's drawing around. And then within that envelope, there are a number of membrane proteins. The big pink balloon-shaped ones that I drew on here, or club-shaped ones, are S proteins. They're actually a trimer. There's 3 polypeptides that work together to make each spike. We've got a protein in blue labeled M. That's a membrane protein. Another protein labeled E, which is an envelope protein. And so those 3 types of proteins make up the surface of that virus, along with the membrane. Within the virus, you have the one really long positive sense RNA genome. As far as RNA viruses go, this is one of the largest RNA genomes around. It's maybe 30,000 base pairs long, and it is associated with this orange protein that I labeled N. That's a nucleocapsid protein. There's a lot more N than that, but we wouldn't be able to see the genome if I covered it up. Anyway, that genome is going to be released when this virus fuses with the cell that it's infecting. This nucleocapsid and genome will be released, and you'll get virus replication started. Switching over to this, the 1918 H1n1 influenza. All influenzas are membrane-bound protein, or membrane or enveloped viruses, which means they have a membrane. So it's just like this. It has a membrane. It also has a number of proteins that are associated with it. Whereas we had a spike protein over here, the main 2 proteins poking out from an influenza virus are the H protein and the N protein. This virus was H1n1, and so this is categorized as an H1 type of protein and an N1 type of protein. And when we talk about the different pandemics of influenza we've had, those will come back into play. Along with the H and N proteins, we've got some orange proteins drawn in here that are designated H2. My dog's wanting out, so hopefully he doesn't scratch too much. M2, that is a channel protein. And then there is an M1 protein, which are all the blue dots along the interior of that membrane. and those are just known as membrane 1 proteins or matrix 1 proteins. Then we have N protein with Spanish influenza as well, and those also associate with the genome of influenza, although you'll notice that the genome of influenza is drawn quite a bit different than what I had done here, and that's because this influenza genome is segmented. There are 8 segments, and a couple of the segments encode more than one gene. There's about 10 proteins in all, whereas over here we've got probably at least 20 different proteins, maybe 25 or even 30. I lost track, I didn't count completely, but there's a whole lot more proteins encoded by this genome than by this genome. Then if you look closely, and if the color isn't faded out, there's a green dot that I drew at the 5' end of, or at the end of each of these virus genome segments, and that is to represent a polymerase. And the reason that the polymerase gets bound up in the virion, the virus itself, is that these RNAs are of the negative strand, which means when they get into the cell, you have to make the positive strand version of that using the negative strand as a template. And those serve as the mRNA for encoding proteins, whereas this was a positive strand RNA virus, single strand RNA. So when that RNA gets in the cell, it can immediately be translated. It acts as if it's an mRNA right from the beginning. Okay, so when we talk about viruses, I've been saying that a good way to think about them is to separate their replication cycle, replication, into 7 steps. We have attachment and entry of the virus, uncoding, so that the genome is available to be used in the cell. Transcription, that's the act of making mRNAs. There's the component production, and once you have enough components, you can assemble them. So step 6 is assembly, and then once you have assembled infectious virions, then those virus particles can be released from the cell. And so for attachment, the protein that SARS-CoV-2 uses is called ACE2. It's also the same protein that the original SARS coronavirus recognized, and the current SARS-CoV-2 has a lot of sequence similarity with the original SARS, and so that's why it was given the name SARS and then just the number 2 added to it. The 1918 influenza, in contrast, it doesn't bind to necessarily a protein. Instead, it binds to sugars that are linked together, and I'll come back to this later, but in humans, it's usually the alpha-2,6 linkage that you connect the 2 carbon of a sugar to the 6 carbon of the next sugar, and so on. And so that's what it uses there. So we've got a protein versus a sugar, a carbohydrate. Next, entry. This entry of the SARS-CoV-2, it could be at the plasma membrane, which is the surface of the cell, or it could be within endosomes. And then in the influenza, it's acidified endosomes. Okay, so when a cell basically takes up the virus and the virus starts to enter into the cell, it's within a vacuole. That vacuole becomes more acidic, and as it becomes more acidic, certain proteins on the surface of influenza will then fuse with the endosome, and the virus genome can get into the cytoplasm of the cell. Whereas with coronaviruses, SARS-CoV, it's possible that some of that fusion is happening on the surface. It's maybe not as much pH dependent, or it could also happen in the endosomes. So that's one difference, is that you might not have that complete pH requirement. Just to stay on that topic for 1 second, there's a lot of talk about hydroquinone. That doesn't sound right. But anyway, a molecule that will block the acidification of the endosomes being as a potential use for this. But if the virus can attach at the plasma membrane, then it won't be that effective against the virus. Uncoding is fusion, either the plasma membrane or the endosome, and that just reveals the genome. The influenza, on the other hand, as the endosome acidifies and you're getting ready to fuse the virus with the endosome membrane, the acid can also make its way into the virion, and that will release the virus segments from that M1 protein that we saw in the previous, and then you have fusion, and all those different 8 segments can be released into the cytoplasm of the infected cell. They then go to the nucleus, whereas the genome stays out in the cytoplasm with this virus. Okay, transcription. There's not an immediate need to do transcription because the RNA, positive sense RNA genome itself is able to be transcribed. You might make an anti-genome and then make more of the regular, then do transcription to get more copies of the positive RNA. But it's not necessary to immediately do transcription. However, when you have the influenza virus, you do need immediate transcription to happen because the genome is negative sense, and so the RNA polymerase is brought along in the virion, and you can make the positive strand RNA virus to that. Okay, component production. Both viruses have a lot of membrane proteins, and so you should automatically, if you've taken my freshman biology class, you should automatically think about these proteins being produced in the ER, and that's true for both viruses. Assembly. Between the ER and the Golgi, there's a compartment of the cell called ERGIC, and that is where the coronavirus assembles, whereas with the influenza, the components of the virus mostly make it out to the plasma membrane, and then the genome ends up getting recruited out to the plasma membrane, the surface of the cell, and then that leads to budding of the influenza virus from the cells, whereas in contrast, these assembly happens within the lumen of the ER membrane, endomembrane system with the SARS-CoV, and so then you actually have vacuoles connecting with the plasma membrane, and you have exocytosis of those viruses. So a lot of slight differences between those two viruses as far as their replication cycle goes. And then just in more general terms, we'll compare just coronaviruses to influenza viruses. Mutations, both viruses get mutations. They're both RNA viruses. RNA viruses are known to have a lot of mutations. That doesn't mean that the virus will evolve or will change rapidly out in nature, because a lot of mutations could be detrimental to the virus. But at any rate, those mutations can lead to something called genetic drift for both of these viruses. So that's an accumulation of little changes at single nucleotides over time. Then moving on to seasonality, the coronaviruses happen every few years, and so it's they kind of have a little bit of an outbreak every few years. Coronaviruses typically are one of the causes of the common cold, and after a few years, enough mutations build up that then you can have another bit of an outbreak. In contrast, influenza virus has a definite seasonality where it gets transmitted around during December, January in the northern hemisphere, and then during what is our summer in the southern hemisphere, their winter. And by the time our winter rolls back around, the virus has mutated enough that then we can have new strains that are able to infect people and get by the immune memory that most people have built up. Okay, so it just takes coronaviruses a little bit longer to go from one to the next. Now, is that true for SARS-CoV? We don't know. CoV-2? We don't know yet. We'll have to see. In addition to just those moderate changes in the genome and the proteins that they make from the genetic drift, you can also have large genetic changes, which in influenza is sometimes called genetic shift. And in coronaviruses, the mechanism for getting large genetic changes are recombination. So it has the one long genome, and if a cell gets infected with two types of coronaviruses, you could have those genomes kind of mix and match to make a new long chain or a long genome. So that's recombination. If you watch my previous video on genetic linkage, that also dealt with recombination. So same idea as that crossing over during meiosis. In contrast, in influenza viruses, the RNA genome is segmented. There's 8 segments. So if you have one cell get infected with multiple types of influenza viruses, 2 or more, then the 2 different influenza viruses can mix and match their genome, and you end up with something called a reassortant virus. And that would be, they take some genome segments from the first and other genome segments from the second virus, and you've got, it's almost like shuffling a deck of cards. It's a new hand of cards. Pandemics. We're dealing with the first known pandemic from coronaviruses, COVID-19. It's possible that those coronaviruses that currently cause the common cold, it's possible those caused pandemics before we had health systems in place to recognize them as pandemics, and now they're just kind of adapted to the human genome, and people have gotten used to them and have the immune system to take care of those seasonal coronaviruses. COVID-19, perhaps it might take that same track and become a seasonal virus. With influenza, we have the 1918 virus, which I mentioned earlier, and it's an H1n1 virus. The 1957 virus, it was one of those reassortants, and then in the reassortant you have the H protein was of type 2 and the N protein was of type 2. So both the H and the N proteins were swapped out in that pandemic virus. In 1968, there was another pandemic, and that was an H2N3 virus. And I grew up in the 70s and 80s, and so like every year our vaccine was against H2N3 variations. So once you have a pandemic, then it just kind of works its way into every year seasonality. And then finally, 2009 was when we had the swine flu H1n1. And so almost 90 years later, 91 years later, we have H1n1 came back. And you can imagine there's very few people left on Earth in 2009 that had immunity to the 1918 flu. So we had a new population that was ready to be infected again. So that's an interesting point there. There are animal associations with these viruses, and with both of these viruses, we have what's known as a reservoir for the virus, and then intermediate hosts for the virus. So for coronavirus, the reservoir is in bats. So if you go out into the wild, you could collect bats, and you can find them carrying coronaviruses. I have a friend, Eric Donaldson, that was doing some of that work. And then the reservoir for influenza virus is in birds. A lot of emphasis is put on like chickens, but the birds in nature that have this are those birds that are migratory birds like geese and ducks and those sorts of things. Then the intermediate host is a temporary host that by mixing and matching with these intermediates, then the virus gets enough of a reassortment or recombination that then it's able to infect humans. In coronaviruses, there's lots of examples. The original SARS was a civet, and then in influenza, it's often thought that pigs are kind of the mixing vessel to go as your step from birds to pigs. Vaccine targets. Right now there's a lot of talk about making some vaccines against, that's my version of the letter S, the S protein, the spike protein. But I put a question mark in there because there is some data rolling out now that say that antibodies that bind to the S protein could make coronavirus worse. You see, antibodies can do a couple different tasks. One of them is neutralization, which is good. That would prevent the virus from infecting our own cells. But the other thing that the antibodies could do is stick to the surface of the virus and make those viruses more likely to be taken up by macrophages. And in that case, you have macrophages taking in the viruses, and the macrophages are big producers of cytokines, and they could make the infection worse. So definitely a question mark on the S. If we do get antibodies to the S protein, we want them to be neutralizing to prevent infection and not the enhanced uptake by macrophages. Then with the influenza virus, the yearly influenza virus vaccines are typically made to the new mutations, the genetic drift that we see with the H and the N proteins of that virus. There is talk of trying to make a universal influenza virus that would either be to some conserved part of the H1n1, which is hard to do, or it could be with some of the other proteins that are made during a virus infection, such as the nucleoprotein or the polymerases. If we have antibodies or if we have CD8 T cells that can recognize those sorts of proteins, they might be able to take care of more than just the current season's influenza. So that video kind of got longer than most of my videos, but there's a lot to compare when you have these two viruses. Tons is known about influenza virus, and the coronaviruses are now being studied much better. But if you have any remaining questions, go ahead and put them in the comments below. And as always, like and subscribe or not.